phosphates, all those head groups. We've got carboxylic acids forming fatty acids, and then we've got now these sphingolipids, and that gets pretty complicated. And so we're going to look at higher structures now that have a lot of different lipids in them, and it gets a little complicated to keep drawing all these details. So we're going to simplify things. So remember, if we have lipids that are amphipathic, like we've been talking about, these contain both polar and nonpolar groups, that we can kind of simplify it down a little bit. So we're going to simplify it like this. We're going to say, yeah, we've got a head group, and then we have either one tail like this that might be nonpolar. That could be something, say, like a fatty acid. Or we could have like a diacylglycerol phosphate, which would have a polar head group, and then again, a nonpolar tail like that. So just label this generally understanding that this is the nonpolar tail here. And then here we've got the polar head groups here, depending on what they particular are. Now we can think about these kind of from a larger scale out, where we can see many of them all at once. So these are very, very sparingly soluble in water, right? We've got this polar group here, of course, will interact with water, but the nonpolar tails won't interact with water very well. This is, of course, the hydrophobic effect. And as a result of that, these don't interact with water all by themselves very well, but they do interact with a phase transition pretty well. And that is where you, say, have water and oil, or water to air. And because of that, we call these surfactants, because they will interact with the surface. You can imagine if you had a water to oil interface, then you could have the polar head group interacting with the water, and then the tail groups here could interact with the oil surface. So I want to look at some ways we can make those sorts of arrangements in different lipid structures. And so the first thing I want to talk about are these things called micelles. And to understand these, what I want to first do is just think about what would happen if we had lipids at low concentration. So I'm just going to, again, use a simple little diagram like this for a lipid. And if I just had a couple of these around in low concentration, water could interact reasonably well with the head group, like hydrogen bonding and dipole ion type interactions, those sorts of things. But if I had another water molecule and it was coming in contact with the nonpolar parts, that wouldn't be particularly stable. And so we just can't really have a high concentration of these lipids floating around like this, such that the water interacts with those nonpolar tails. So at low concentration, it's okay. There's a lot of strong interactions going on with the head groups that the solution can tolerate some interactions with the nonpolar tails. But I want to contrast that with the high concentration situation. So if we have a lot of these lipids, these amphipathic lipids in water, they can't just swim around like this. They're actually going to start to form higher order structures. And that structure is called a micelle. And it looks like this, where the non-polar tails end up kind of buried in the center of this structure. So I'll just draw a few. You don't have to get the exact same number that I do, but the idea here is that we have the nonpolar tails buried on the inside of this structure, and then the polar head groups are all on the outside of this structure. Kind of like how we looked at how proteins fold up and put the nonpolar side chains on the inside and the polar side chains on the outside. So just I'll point out, this is actually a sphere, but I can't obviously draw it as a sphere on the screen here. But it's think of this as a two-dimensional sort of cross-section of a sphere. Okay, and so what we would observe then is that water now, right, can interact with the polar head groups. So let me just write polar head groups are on the outside, and so if I'm a water molecule, I'm totally cool with like hydrogen bonding or having, again, those ion dipole interactions there. So water can easily interact with this structure, no problem. And then the nonpolar tails I'll say NP for nonpolar tails, are inside this structure, and then they don't have to interact with the water. So this is a very stable arrangement for lipids at low concentrations. And again, this is a two-dimensional cross-section. Well, how does soap work? We talked a little bit about soap and surfactants and detergents and those sorts of things. Well, you can imagine if there was, say, an oil droplet, and I'm just going to say oil droplet looks like that. Say it's on your hands or on your clothing or wherever, and we want to wash it away with water. If I just try to wash this away with water as is, that's not going to go anywhere because it can't interact with the water. But if we had a soap molecule or other surfactant, detergent type materials, the same sort of thing can happen. It can almost form a micelle around that oil molecule or oil droplet. And now we have a structure that would look something like this, where now water can interact with those polar head groups, and then this can essentially be dissolved. So this makes the oil dissolve, even though it's not really dissolving in water. It's using an adapter molecule. I think I've used that term before. So in the sense of using soap to wash our hands, we're using soap as an adapter molecule to make the sort of transition between nonpolar and polar. And then finally, what we're going to move into next is the lipid bilayer. So rather than forming a spherical micelle, we can form a lipid bilayer, which is a stable arrangement of these sorts of things. Again, the nonpolar tails remain hidden from water. So you got the nonpolar tails in the inside here, and then you've got the polar head groups are exposed to water, and that of course is a stable situation. And just to point out, right, we would have water on this side, water on this side. This could be the outside of the cell. This could be the inside of the cell. We could have, maybe it's an organelle inside the cell, like the mitochondrion or something like that. We'll talk more details about that sort of thing. This is what we call compartmentalization. It enables us to form distinct spaces within the cells or within an organism. So we could make distinct spaces like the cell. Without a membrane, there is no cell. All the contents wash away and we have nothing that we can call a cell anymore. And within the cell, we have organelles like the chloroplast, like the mitochondria, like the nuclear envelope, those sorts of things. That's what these are made of.